Welcome to today's HPL seminar. For the next hour or so, my name's Ashna Gumbari, the HPL seminar moderator. Today I'm introducing Mao Delgado, who's a PhD candidate here at the University of Calgary, with Dr. Walter Herzog. Mao completed his Bachelor of Science in Kinesiology and Physical Therapy all the way back in 2007 in Metropolitan University of Educational Sciences in Chile. And then in 2020 completed his Master of Science in Kinesiology from the same university. And now he's a PhD candidate here in the university with Walter Herzog, and his research topics are obesity, metabolic syndrome, osteoarthritis, and biomechanics. So for an interesting background on him, he gave like 7 here, but we'll save a couple for his next seminar. He wanted to say he's the oldest trainee, he says, in the HPL. He's a musician, a bass guitarist, and has a rock band, and apparently you can find him on Spotify. And he plays music too. Oh, Apple Music, sorry. Spotify and Apple Music. And he also recently became a grandfather to 6 baby rats. With that, I'll give it to his presentation titled The Potential Predictive Role of Intrinsic Aerobic Capacity on Obesity-Induced Muscle Degeneration.
Thank you, Mike. Thank you everyone for being here today. Since my last seminar, I think it was 1 year ago, and I finished that seminar saying that just bear with me because we were in the beginning of our project in that seminar. So now I have interesting results that we can discuss and maybe address other questions. So as a physiotherapist, I am a physiotherapist and I have clinical background. I always remember that being worried about the impact of all the health issues that our clients, patients has. And it's not just about one little thing, could be others that are masked and hide from even me as a rehabilitator. So being in this project with Dr. Herzog has been super interesting because basically I am facing that reality again to address the whole impact that it has, the obesity and the metabolic syndrome that could lead different consequences. So for example, the muscle degeneration. So that brings us the idea to say that the muscle can be detrimental, affected by the age, of course, diseases, injuries, and the lifestyle. And the lifestyle will be focused specifically in the nutritional part, and of course, the physical activity. So it is important to mention that the obesity is part of the whole metabolic disruption in our bodies, that we call it metabolic syndrome, okay? So I think it's easier to connect with the idea that the obesity is just the only thing that we have, but no, there is more things that are involved, and we can cluster it as a combination of those effects in our body due to this metabolic distress that produce, for example, the high fat reduced diet that we are super, related every day, no? So hyperglycemia and insulin resistance, dyslipidemia, hypertension, and diabetes, for example, could be some presence of this, or can mention that presence of the metabolic syndrome. So if you have 3 out of these 4 or 5 conditions, we can be sure that you are under the presence of this metabolic effect. Doesn't mean that automatically you will be gaining weight. You could have this condition without any change in your body. So that is important to mention. Of course, obesity has been linked as different impacts in several systems among our bodies. And of course, those are associated with several comorbidities. So it is hard to distinguish who is first, what is the most important one. So for example, in our clinics work, we approach our patients with this multi-systemic background affection, no? So in the obesity rehabilitation, we work with a multidisciplinary team that address not just cardiovascular condition, even liver disease, also cancer, diabetes, but me as a physio that are focused in musculoskeletal rehabilitation also, I want to talk about the associated musculoskeletal disorders that we are seeing in our experiments, and specifically due to the increase of the mass and therefore increasing the joint load, but of course, focusing specifically in the metabolic syndrome and the metabolic OA like that we are studied with others trainees in our group. Okay, so I will be focused specifically in the metabolic syndrome. I won't touch anything about the load increase in our joints. So let's go into. So previous work from our lab showed that metabolic syndrome leads to this development of musculoskeletal degeneration in mature rats exposed to high-fat sucrose diet. So has been a really prolific line of research in our group, and I am super happy to be part of the second part maybe of this big, maybe 10-year project. Okay, when we talk about musculoskeletal impact, researchers has been demonstrated that the cartilage could be damaged due to this metabolic distress. But I will be focused specifically for my first study, my PhD, about the impact in the muscle integrity that leads this degeneration of metabolism. So a high-fat sucrose diet includes these muscle changes, and all the evidence that our group has been found lead us to the idea that this impact is very muscle specific. So what does it mean? That the changes in skeletal muscle metabolism occurs specifically depending on the type of the fibers that those muscles has. So all research has been demonstrated the muscle like vastus lateralis, for example, that it has a higher proportion of type 2 fibers with a metabolic glycolytic metabolism, showed an increase in pro-inflammatory macrophages, increase in the muscle fat infiltration, muscle fibrosis, and a decrease in the muscle mass, a phenomenon that we call it sarcopenia, obesity sarcopenia. Okay, so interestingly, all the research that has been done in our group haven't seen any significant change in muscle like soleus, that is a high intrinsic aerobic metabolic muscle. So the question that brings us the idea of, does the inherent aerobic metabolism could play a role here? So that is the first main question that Dr. Herzog start to explain to us, no? So the idea brings us to say that maybe the aerobic capacity may not only protect against this metabolic disease, but may also protect musculoskeletal tissues from degenerative changes in the presence of obesity. What is aerobic capacity? So aerobic capacity is a combination of several factors that will show us our oxygen consumption. At the end of the day, we have a genetic background that gives us around the 50% of this aerobic capacity. But of course, environmental factors could lead to this final aerobic capacity that you can achieve in several tests that were very popular here in our research. So how we can achieve this aerobic capacity in our models that we are interested in? We can train our humans, of course, or train our W rats with a specific aerobic training to increase that aerobic capacity. But for that condition, we name it acquired aerobic capacity. But some of you maybe know that we can select breeding group of rats to isolate the intrinsic genetic background to just show it and highlight it, that aerobic capacity that came inherently from the genetic background, okay? So our collaborator, Dr. Lauren Koch, in 1996, started to create this selective breeding system from 2 lines of selective ovary rats. And she matches two lowest and the two highest performing rats to mating for different several generations to start to isolate that aerobic inherent condition. And they repeated for the same 3 generations. So basically they tested the rats with a evaluation of the running performance. And then basically they matched the two lowest performing ones, generation after generation after generation, to get a specific strain of rat that we call it the coach potato rats, or the low capacity runner rats, okay? In the other side, they did the same with the two highest performing ones to get this amazing high runner capacity that is some of you maybe know, yeah? So now we have this very selective group that without any training, just with the matching, outbreeding, they get these capacities in a low and in a high way. So let's see how they develop this aerobic capacity. After 3 generations of outbreeding, the offspring showed after 3 generations and a change of 70% of the distance that they run. So we can see that difference there in the graph, no? What's happened after 6th generation? 171% of difference between the highest and the lowest performance. After 11 generations, 347% of difference between the highest and the lowest. After 28 generations, around 800%. So, and that brings us the idea of what will happen if eventually we keep matching those rats. We will see what happens after 49 generations. Okay, so at the end of the day, studies have been shown that those rats has an intrinsic aerobic capacity that the most important explanation came from the metabolic aspect in general. So specifically, we can mention that the mitochondrial function of those rats are totally different than the low capacity ones. And evidence has been investigated to see how these rats could be affected by the high-5 sucrose diet or the presence of metabolic disruption could be totally different. So investigation has been shown that those rats, the high capacity ones, showing an increase in the cardiovascular function, less weight gain during this high fat diet intervention, less metabolic complications, less diabetes, and an increase in the 45% in life when we are compared with the low capacity ones. So that I'm sorry, I think I skipped one slide. So, but there is no research in those outcomes that they explored sort of musculoskeletal outcomes. So the idea is to translate or investigate if we can translate to partial or full protection of the degenerative process of the musculoskeletal system due to this high-fat sucrose diet. So the question of my first study is the inherent aerobic capacity protective against metabolic syndrome and musculoskeletal degeneration in the presence of diet-induced obesity? So the hypothesis for my first study is that the aerobic capacity protects rats exposed to a high-fat sucrose diet from this musculoskeletal degeneration. So the big objective, the main objective of this study is to test if the inherent aerobic capacity achieved through more than 49 generations of selective outbreeding protects the musculoskeletal system from this degeneration in this condition of diet. So I remember that I asked the same question in my first seminar. Some of you were here, and I think Mike nailed it with the answer. So I asked it to you. So can you imagine the difference among 49 generations of breed? Do you remember what is that name? Large, you're not participating? 1,000, more or less, no? So this is our rats. After 49 generations, the high capacity ones in this test, or test the distance that they run is really, really different, no? So we know that we bought the right rats, no? Because they are very expensive too. Okay, so we use the same protocol that has been proven that works to produce these metabolic changes. So 36 male rats with a diverse inherent running capacity from 49 generations. So we use the half of high capacity runners and low capacity runners with 25-year-old age. So we divided in two groups. The high capacity runner with high fat sucrose diet. The diet basically is 50% of fat, 45% of sucrose. So very, very yummy food. They love it. And the same group, high capacity runner with a chow diet, regular pellet diet. And the low capacity ones with the same diet. And at the end, we have 4 groups to investigate the effect of this diet. So for 12 weeks of diet intervention. We collect several measures through the whole timeline of the 12-week interventions. So this is a summary of all the analysis and all the information that we recollected. So we did an oral glucose tolerance test, blood collection, DEXA scan, feces collection, food intake, and body mass tracking every single day. And because there is other outcomes that they are interested for Dr. Rainier group, so basically it's a multidisciplinary study, no? I won't present the most of them, so just I will be focused in this outcome that I will present now. After the harvest, we took some sample, the whole muscle, vasculateralis muscle and biceps brachii muscle from our rats as an example of representation of fast glycolytic muscle, okay? And we took the soleus muscle as the biggest representation of the slow twitch oxidative metabolism. How we investigate the muscle integrity, because that is the big topic and it's important to define it. After to check the presence of metabolic syndrome, that is important to say that, okay, the diet is working. We collect the muscle, or we investigate the muscle fat infiltration, the muscle fibrosis, the muscle inflammation, and of course, the function of the muscle that we call it muscle mechanics. And we use it histologic analysis or investigate those outcomes. We took 10 micrometers muscle sections from just one whole muscle from a rat, soleus muscle. Look at how big it is, really tiny. And we mounted in this cryostat machine to cut it some sections and prepare our slides for this histologic analysis. This is a video that it was basically 3 months cutting muscle, so I am very proud of this process, so that's why I'm bringing it. So then after this process, we stain them to investigate the presence of fat with the all-rate oil staining process. We proceed to rate the stain for collagen and enzyme isochemical David reaction for the data effects, okay? And after that, we use a combination of imaging and math script to analyze content and finish our analysis of those images. For the mechanical test, we skinned muscle fiber from soleus and BL, and we mounted in our mechanical testing system. And we calculated, or we tested in an average sarcomer length of 2.4 micrometers. And the outcomes that we took for that area is the passive force among those distance in the sarcomer, the active force at 2.5 micrometers, the shortened velocity, and the calcium sensitivity and covert. So let's come into the result. Just an DEXA scan image to just see how they look. So we can see a subject, a rat from the high capacity runner rat with fed with high fat sucrose diet. And they are smaller rats and they didn't gain too much weight in the way that they guys, the low capacity rats with the same diet. So you can perceive some difference, but of course, we measure the body mass through the whole weeks, and you can see how those strains are completely different from the beginning. But interestingly, and this graph here came from the last week of intervention after 12 weeks, we can see that the high fat sucrose diet slightly increased the body mass, but we didn't see any significant difference as we saw between the strains, okay? So we were very worried about what's happening with the diet. So it's not working, but it is working and we can see it in the body fat content. So look at how the high fat sucrose diet affect the both group of strain. So basically, the high fat sucrose diet or low capacity and high capacity get affected, and look at how they look after the third week of diet intervention. Both in the same way they increase the body fat content. So the diet is affecting or is doing something for both groups. What's happening with the muscle weight? Bicep brachii, vastus lateralis, and soleus, we didn't see any significant difference in the weight when it's normalized by the body mass, when it's normalized for the size. And we can see just one significant difference in the low capacity runner ones, just in the vastus lateralis muscle, okay? So those guys that they were fed with high fat sucrose diet, they were affected, they decreased the muscle weight in comparison with their body weight in a higher weight than the last high capacity runners does. So I want to mention that the order of the graphs will keep the same idea, bicep, BL, and soleus for all the slides, okay? What's happening with the oral glucose theta and insulin sensitivity? So look at how the diet affected the glucose content after this 1 hour test that we were exerting that time. So both groups, high aerobic capacity and low aerobic capacity under the high fat sucrose diet, increase the amount of glucose. So again, the diet is affecting both groups. And this is a comparison from the beginning, no? From the first baseline. The HOMA IR index shows the insulin sensitivity or the insulin resistance, and something interesting is happening that just the high capacity runner one under the high fat sucrose diet express this high insulin resistance, showing a sort of metabolic disruption already. Not like the low capacity runner ones, because we were expecting something totally different in this way. So keep that in mind, that it will be important at the end. So let's go for the fat infiltration. This is an image from the vastus lateralis, and you can see I marked it in red arrows, the presence of fat. And this is a comparison between the high capacity runners with the high fat sucrose diet and low capacity runners with the same high fat sucrose diet, okay? So that is the area, vastus lateralis. And look at how the amount of fat increased in the high capacity runner. Totally the opposite that we were expecting compared with the old results from Brown W. Okay, so increasing the amount of fat. And look at how the graph, of course, showing the same bicep, no significant difference. But look at how those high capacity runner ones storage more fat even under the high fat sucrose diet person. Yeah. Another interesting result is the no significant difference in the soleus mass. So this is super important because it's in line with the same result that has been demonstrated or group that brings us this idea and this whole project that maybe the aerobic capacity of this muscle could be protected already inherently And we didn't see any significant difference in this group. What's happening with the muscle fibrosis? So decrease in collagen content. Again, just in the vastus lateralis. High fat sucrose diet for both, the same idea. We are seeing here the same process for the collagen among the ages of the fibers. So we are able to count it, and interesting result we found. So bicep bracket. So look at how the high fat sucrose diet increased the amount of muscle fibrosis in both groups, but even higher in the low capacity ones. So the diet is affecting both, but in a lower way in the high capacity one. Super interesting. Vastus lateralis, or favorite muscle. Look at how just the low capacity ones are increasing the amount of collagen content, and no significant difference in the high capacity runners. So we are seeing some of the protection that we were expecting eventually. And again, soleus muscle, no significant difference again in the presence of the high fat sucrose diet. Is slightly trained to increase, but look at how in the same way the slightly decrease for the high capacity one. But I think it's fair to say that no significant difference in the same way that we were expecting. What's happening with the muscle pro-inflammatory macrophages? We are seeing the same idea, the same BL muscle, vastus lateralis, and I am marking the presence of macrophages, pro-inflammatory, M1, that they are triggered the inflammation process eventually, and could be connected with the other outcomes that we are analyzing. So we can see how the most of the images that we see are pointing out those macrophages in the track of the collagen that are there, but sometimes we were able to see some kind of crowns of macrophages that try to phagocytate a whole fiber, but this wasn't too common. This was the most common representation of macrophages. So that is the idea of this staining process, and the results show we don't have yet the biceps bracket, so even for the next seminar maybe. So just I bring the vastus lateralis and look at how the presence of inflammation, it is higher in the low capacity runner ones compared with the high capacity ones under the presence of high fat sucrose diet. So, and what's happening with the soleus? No significant difference within the diet, but there is a factor, an effect of the strain that show it, and when we put all together, those rats show it a lower average amount of macrophages compared with the low capacity rat. There is no effect of the diet here, but we can mention it that impact. We know that we have an outlier there, but at the end of the day, we can say maybe no significant difference either. So from this skinned muscle fiber mechanics, no significant difference in all the outcomes that we explored. So that was among all the groups. So basically, we are ready to discuss some concepts that we are considering to keep it for the next project. So in terms of the muscle fibrosis and the inflammation, so the interesting part was the only fast glycolytic muscle as biceps brachii and BL were affected due to the obesogenic diet, but not the slow oxidative muscle. So that wasn't the trend that we were expecting and the hypothesis that we were aiming for. So the increase in collagen content in the vastus lateralis, in the low capacity runner that could be lead from the diet effect. But the high capacity runner, they didn't show any significant increase in the in the fibrosis condition. So we can maybe arise the question about could be an protective effect against this fibrosis in those muscles and in those group of rats. Evidence has been shown that there is a strong relation between the metabolic distress, the inflammation, and the fibrosis in muscle. Remember that when we have a metabolic distress in our muscle, the natural consequences is to start with the inflammation and repairing process eventually. So that brings us the idea that maybe the presence of fibrosis could be related with the inflammation that came from the macrophages activity. But that is something we have to explore even further with some relations and correlations that we have to set. In terms of fat infiltration, graded fat infiltration in BL, but that was something that we didn't expect in the high capacity running. But interestingly, we were discussing, okay, so what is happening with those muscles that they don't have this aerobic capacity? So maybe the high fat sugars diet are showing us something that in humans we already know, that it is the athlete paradox. So basically, the thing that is happening, so these rats, eventually they are storing I think in a more efficient way that available fat that is there in our tissues. And that is something that is very similar that happened with the athletes that we are investigating around the world. So what is the athlete's paradox? So highly aerobic trained human athletes, they show higher content of muscle fat infiltration, but without manifesting the muscle metabolic change, okay? Such insulin resistant or others. But the difference here, the biggest difference at all rats is just a strain that came from an inherent aerobic capacity. There has been no training at all. So maybe it's an unfair comparison because marathoners like those guys are training every day and maybe they are consuming that fat every day. But those rats, all rats, are not in that case. They are basically trapped in a small cage all their life. So basically, that is something that is important to keep it in mind. So maybe that could be aligned with the presence of metabolic disruption that we are seeing in the insulin resistant, for example. Okay, so the evidence has been showing that this high capacity rat has an enhanced mitochondrial content and function. And the researchers propose that this is a mechanism of health protection. So when we go into that papers published already, they show it that those high capacity runner rats has a greater amount of mitochondria. Okay, so that is the biggest difference. And that's explained the higher oxidate consumption that all the research has been tested at rest and during exercise. Okay, so they showed an increase in RNA and protein expression, and an increase in the maximal phosphorylated respiration, and less oxidative stress damage, with a higher fatty acid oxidation, lead us to a decrease in the respiratory quotient arrest. So, I am not an exercise physiologist, but that I think is important to keep it in mind, and that brings us the idea to maybe think to explore some of those out of that could give us some idea. So I don't know if you remember this image that maybe was a little bit tricky and complicated in your past days, but this image showed the oxidative phosphorylation process that basically represents the way that our mitochondria in the inner membrane layer produce pump electrons in the electron transport chain to produce at the end of the process ATP. No? So this is the biggest machinery to produce ATP. So if we remember how this process works, it uses the fuel that is available like glucose or fat. So eventually, maybe the next step is to start to investigate the changes at that level to see if the diet is affecting those. So for that, basically, we bring the idea, okay, let's do it. Let's do it. So basically, I started 1 month ago to start my Western blotting of proteins that came from those complexes to demonstrate first, if those rats, the high capacity rats, has this enhanced mitochondrial function, but most important, to see if they get some of protection against this high fat sugar diet at this level, okay? Those results that I will present now are preliminary, just the half of the rats I tested in the vastus lateralis master. I didn't calculate any statistical analysis, just to show it slightly trend that we are seeing. Okay, complex 2, complex 3, complex 5. Maybe the most important one, if you can say that, is the complex 5 that is producing the ATP at the end of the day from the fuel and all this machinery of electrons that are moving those protons. So look at how, in general, the trends show it that all rats, the high capacity ones, are slightly higher in those proteins. So this is not a direct measure of the mitochondria content, but we are measuring the expression of the proteins that came from the electron transport chain, and it's really good correlation. There is a really good correlation between those proteins and the efficiency of the mitochondria and the presence of more mitochondria. Okay, and I don't know if you can see, but I don't want to be too optimistic, but maybe some complex we are showing some of those changes due to the diet. But that is something super interesting to keep working in the future. So stay tuned for the next session, please. So as a conclusion, we confirm our hypothesis. The increased aerobic capacity protects animals exposed to a high-fat super diet from this musculoskeletal muscle degeneration, specifically in the muscle fibrosis. But of course, further investigation is necessary to understand how an obesogenic diet impacts the metabolism of different muscle types, and whether the increased aerobic capacity can offer this protection at this level. So my future directions are to complete the mitochondria analysis, the OFOX analysis, to include other markers of metabolic stress in muscle, for example, the glucose transport and muscle insulin resistance, and to explore structural and functional muscle proteins and myosin, actin, titin, and neolin. And of course, something important that we need to address too is investigate the potential impact of the high-fat super diet in the fiber type proportion in our muscles. We have some data that confirm that those muscles has those type of fibers that we were expecting, but we need to do more analysis to be confirmed, to be sure that we are analyzing the right type of fibers. I want to acknowledge all my supporters, my group, my amazing group, my agencies that found and let me to be here with you. And thank you so much for your attention.
Very thoughtful questions.
Laura, thanks for that great talk. I'm curious, you showed that there was greater fat infiltration for the vastus lateralis, right? The high capacity rats. But you briefly mentioned that there was no change in muscle function.
Yeah.
So what do you think is going on there? For example, any change in function?
Yeah. So that is a question that has been around since forever, because we started with the muscle mechanics outcomes. And to be honest, just speculating, I think the impact of the diet, it is really mild. Okay, so under 12 weeks of diet intervention, maybe we are seeing just some level of changes, but I think a longer exposure to this type of diet could lead us to see some functional changes. And I know that it's hard to translate this to humans, but in humans, we can see that the obesity producing more patients and increasing the force first. That is natural outcome. But then, after years of metabolic syndrome and obesity, that force change when you normalize for the body weight. And at the end of the day, they show it and a decrease in the force production. We started with the skin muscle fiber. So now it is important maybe to address other muscle levels. So maybe the whole muscle and maybe other type of evaluation to see if there is some change at that level.
So do you have a genetic background of the food?
I'm sorry, I didn't.
If you have the genetic background to,
The 49th generation of the rats? We should have it. In our collaborator, they has been done all the tested, and there is evidence that shows that genetic background. And it is available that show it where is located that differences. So we are not now using that data, but eventually we can reach out to our collaborators, and of course, they have all that information.
if it's related to mutation, it's better to know genetically.
Sorry? Can you repeat it?
If something related to mutation, it's better to know genetically.
Yeah, sure. Yeah, that is maybe another future direction. Yeah, for sure.
I'm gonna ask, on the slide where you show the population or the sample size, you, for the high sucrose diet for the, high capacity rats, your population says 10-1, is correct?
Yeah, one passed away at the end of the intervention.
I was curious what the timing of the start of this diet was for all of the rats. You said it was a 12-week course. And I guess depending on your answer, do you think whether the diet starts during adolescence or during youth might have an impact on muscle growth and
Yeah.
fat infiltration? Is there evidence one way or the other for humans?
Yeah, good question, Adam. The age has an impact. We have data from the early rats, juvenile rats. And the thing is, they don't show, they start with some detrimental effects due to the age, but of course, those outcome became worse after the adulthood. So basically, they don't show it significant changes when you compare with the adults. They are more protected. Now we are in the final analysis of the last group of those juvenile rats, but we are not seeing significant difference if you start seeing when you are juvenile. Yeah, but the evidence try to show it that could be something that you have to address because the time that you give that, of course, and the age and the combination of the age effect could lead us to the degeneration, muscle degeneration. So in this case, all our rats at the same age, so we try to control that outcome. But eventually, of course, all the rats, they will show it even lower or worse outcomes from muscle. Yeah.
Our nice presentation, kind of building on Colin's point about the muscle function. If muscle function wasn't affected in both strains, like high aerobic capacity or low aerobic capacity, can you confidently say that high aerobic capacity protected the muscle?
I think yes and no, because we were discussing with Martin McKean, Dr. McKean's group, because of course, we need more insights from the exercise physiology part, that eventually that protection is at the beginning of this diet, this intervention. But the question that I have now is how long they will be protected. We can say that under 12 weeks are already protected, at least in the fibrosis outcome. But maybe if we explore to extend on a longer diet intervention, we will see some of damage eventually, because these rats are not being trained at all. So that is the question, because we are seeing a higher insulin resistance in those muscles. But the good thing about it is, okay, they increase the fat that is stored, yeah, and they are showing just preliminary a slight increase in the mitochondria function. So maybe, just speculating, maybe in those 12 weeks, that amount of fat that they stored, they were consuming it to basically produce energy, And maybe, speculating again, that could lead us to sort of protection. that enhances mitochondrial function that is using that fat as a fuel. Because we know that humans has been trained in endurance, they can use the fat in a better way, I think. So maybe they are consuming that fuel in that period of time. And that could lead us to that sort of protection that we are seeing in that window gap. Okay, something important to mention that the fibrosis and the amount of collagen, if you remember the graph, we are not seeing 50% of fibrosis, 60%. We are seeing really slight increase of fibrosis. It is a significant difference, but we are under the 10% of collagen. So we are still in the lowest values of fibrosis or collagen content. That is very muscle dependent. Other muscles like pectoralis that other trainees are working on, they show it more fibrosis, for example, because they have more collagen already. But the increase is around 5–10%. Yeah.
I think you kind of just touched on it, but I was gonna ask if there is any research on this in non-locomotor muscle groups.
Good question. I'm not aware. We were trying to find evidence in other muscle. There is evidence in gastrus muscle that show it kind of the same, but there is not studies. We are almost the first study investigating those effects of the high-fat sugar diet, but I am not aware of that. They use it, yeah, mostly locomotion muscle.
It's kind of related to that. Would you say it's worse to have fat infiltration or more collagen? Like which one's worse to have if you had to choose one?
I think it's important to say that it's maybe bad if you are super sedentary, like me, for example. Because basically, I think when you start to train, it's not just about doing exercise, it's about how you manage the fuel that you are consuming, no? So that is something that you have to address. And I think the ability to burn that fat, say it's simple, it is the way that we can see that paradox in high performance athletes, that they are storing easily the fat, intralipid fat, okay? So intracellular fat could be slightly different than the fat that we are measuring. But that ability to store it, they are showing us that they are not producing any metabolic impact in those. So it's not just about the amount of fat that you are storing, it's how you are consuming with exercise. So that is I think.
Oh yeah, good presentation, Arisa, thanks.
Thank you.
You were mentioning that your high capacity have more mitochondria.
High capacity,
Yes.
that's better
Or
mitochondria.
better mitochondria. But I was wondering if that could be also that the rats have more apoptosis or more cell deaths, and mitochondria has like some effect on apoptosis, and if that can explain some of your results where you're not seeing what you were expecting on this high capacity, because there's more or better mitochondria can give you more like an increased oxygen, but can also, if something triggers, can lead to more cell death. I was just, I don't know, maybe something.
I think I am not in the position to answer that
Okay, no,
question.
it's, yeah.
I am just starting as a biomechanist. All my projects shift to more metabolic aspect, and then now I am trying to go into more and more. But because in the way that we are analyzing mitochondria, it's maybe the most simplest way to
Yeah.
test the function and the quantity. Yeah, we are not measuring with electron microscope, but it's an indirect measure of the presence of more mitochondria. And evidence shows that exercise, of course, produce and increase the amount of mitochondria available. So, but that is, I think, at the entry level for
Yeah.
this type of analysis, and I think we will need to incorporate more insight and people with kind of more ideas that could give us this type of answer. I'm sorry.
No, we'll talk. Thanks.
Thank you for the talk. I'm curious, I don't, I didn't catch this, but, I wonder if sex differences, would be something that has been studied or something that you would look into for, like, the topic of fat storage and utilization, because I know, like, in humans, for like endurance athletes, males and females obviously have different, like, breakdown of fat and how that works at, like, the higher, endurance levels.
Great question, great question. Yeah, that is the plan for the next project, and I think you are giving the chance to talk a little bit about the next step. But that is the plan, because we are seeing difference due to the sex. So the sex has an impact due to this way that they store the fat. And our papers has been published. Hannah Smith was the last one that published those difference due to the sex. So I don't have it in my mind now what are the trends, but of course, I think the female store more fat in general. But that is something interesting to explore. What is the distribution of that fat and what is the type of that fat, So, but yeah, eventually in the future, Dr. Gerson in our group wants to explore the idea of, okay, let's incorporate to our study sex differences. So now we have just one female. I don't know why, because we ordered just male rats, but surprisingly, one turned that it was a female one, and it is true. Now we have 6 pups in our colony, but that is all there is.
Nice talk, Mauricio.
Thank you, Ines.
And maybe I missed it, but did you talk about the difference between the biceps and the VL in terms of fibrosis? Because it seems that VL did not show fibrosis, but biceps did. So why do you think that? And they are both like in the high capacity runners, and they are both.
It's just to, maybe I will bring it up again, just to. Fibrosis, no? So we are seeing an impact of the diet in both groups when we observe the bicep brachii, but not in the VL. My only answer for that is now I want to explore the fiber type composition. I think maybe we cannot match and say that vastus lateralis and bicep brachii has the same proportion of muscle fibers type of different types. So I think that could be an sort of bias in our results that we have to address. So I mean, we are working on that to try to know what is the proportion, the real proportion and amount, or what is the type of the fiber. So I think that would give us some ideas about those differences between the muscles that we are seeing.
So what do you expect, like,
Okay, it was super interesting because there is not too much research about the fiber types in bicep in rats. So everyone is studying the kidneys. So we were looking, but good. Yeah, nothing from pectoralis and biceps. So my speculation is maybe it is more like a mixed type of fibers.
It's usually like 55 to 45.
Yeah, so we are expecting something like that to see, maybe 50% glycolytic, 50% oxidative, but that could bring us other thoughts, because if they increase the amount of oxidative as low, why they are not showing up this sort of protection. So, or maybe it's totally the opposite, and they have even more glycolytic fibers than the VL in proportion. So that is something interesting to address.
but there is a transition from fast to slow fibers. Slow fibers are known to have more collagen around them.
Here we go. Yeah. Thank you.
I just had a question, thank you for your talk. I just had a question about the breeding. Do you think that eventually there will be like a plateau in the differences between the two strains as the generations like keep going, or do you think it's just gonna keep get bigger?
Good question, huh? So now our regeneration are 55, maybe 53, 53, 52. I am expecting to get some plateau in some point. They are mammals, so but I don't know where, but I would love to see it when it will be done. So, yeah, 10 times the difference is a lot. So, but I am thinking that it will be a some plateau eventually. I don't think that it will be like a non-stop difference.
So it's the high capacity runners that are getting better. It's not the low capacity that are getting worse.
No,
Is that
yeah,
the
exactly.
outside
Yeah,
bit?
they just basically get the lowest performer level and they keep it there.
They keep it.
Yeah, because they are running, so you will have some distance to run.
Okay, yeah, so that's the minimum. that
Yeah, the
they
minimum that you
can
can run at that
do.
level, at that
Okay.
pace. Yeah, so they just keep it that level all the time. And I would love to see, but okay, because our next project is now, we will do exercise intervention to all the rats to see if the exercise and the training, aerobic training, could change those effect that we are seeing in those low capacity ones. So that is super interesting because at the end of the day, if you think about how to translate this question to the human research field, basically, we don't have this type of high performance that they were matched together. No, we cannot do that. But of course, the most of the population, they look and behave like the low capacity runner, maybe. And it will be so interesting to see and to say at the end of the day that just with exercise, we can achieve that sort of rotation. And you don't need to have this genetic background, or maybe it's not too important. Or maybe it's important, but that is a lottery. So start to do exercise before you decide to start with this bad diet. So that is, I think, the most important aspect of the second project that we are starting this year.
For the running assessments, are both groups running at the same speed?
Yes, the running tested, they go to with a 50 degrees degree of inclination, and they run at the same speed until the exhaustion.
So, what if you adjusted it so it's relative to a certain value? So that way you can match, like quote unquote, for fitness level.
Yeah, they did that too, and there is another strain that they developed, the same group of researchers, that they call it intrinsically inherent training. Or basically they train them to demonstrate that they can lead some sort of rotation under exercise conditions. For the training part, we will, of course, we will have to normalize and put this match to, depending on the running distance that they achieve. So we will test it, and then we will have the 80% of that distance to match it all the runs in the way that you are mentioning. Yeah. Yeah, that is the way for the training part.
Cool. There's no more questions. We'll thank you all one more time.
Thank you.